who has founded and directed the Boulder Chickadee Study that improved understanding of how humans shape the evolution of backyard songbirds. In her talk tonight, she'll dig into how she leveraged citizen science and built a framework to bridge the gap between scientists and communities to help explore hybridization in Colorado chickadees in an urban world. So please join me in a warm welcome for Kathryn as she presents a tale of two cities, Cryptic chickadee hybridization in an urban world. Kathryn.
Hi everybody, thank you for that lovely introduction, and it's really great to see all the incredible events that you guys have planned. So my name is Kathryn Grabenstein. I'm currently a Rose postdoctoral fellow at the Lab of O, but I've been involved with the Lab of Ornithology and been in Ithaca for a lot longer than that. And today's talk is going to be, I took a lot of artistic liberty, so I hope you enjoy it. It's going to be less science and more how I as a non-birder, I wish I was a birder, you guys seem like you're a lot of fun. I as a non-birder came to be in front of you studying birds. So it'll be a little bit of personal path. We'll sprinkle in some science. We're going to hit almost every major region of the United States along the way, which is, I guess, to show you that I just am really far flung and have been in a lot of places. So I introduced myself as an evolutionary ecologist. It means I like to look at how species interact in an evolutionary framework, and I'm really interested in understanding how those species interact with What else do you want? There's a lot of tech happening up here that you can't see. But there's like 7 plugs in my computer, one of which almost took somebody out, so. Okay, are we good? Anything else? Yeah? Okay, you can hear me, you can see me. Everyone here can hear me, see me. Okay, great. I like to study how species interact, and I like to do it in an evolutionary framework, and I'm really interested in how us as people are changing how these species interact. These days, I study hybridization and speciation, but if you ask my mom, I study birds. Oh my gosh, it's gonna miss. Okay, I want to, there we go, okay. I'm also a mentor, a teacher, an advocate, and a storyteller, and my favorite part about being a scientist is that I get to do all of these things at the same time, or little bits and pieces of them. And today is really going to be about how I kind of leveraged all these different hats that I wear to build the Boulder Chickadee Study, which at the time was just a dream of mine. And now today is something that is highly productive, full-fledged, and really a joy to continue working on. So I told you we're going to do a little bit personal past. This is me. My story starts in Chicago, all the way at the tip top. So I grew up in Rogers Park at the time, and I think still it was the most diverse neighborhood in Chicago. So Chicago, if you've ever been there, is typically pretty clustered by ethnicity. So it was a working class neighborhood. And the key points is that it was pretty concrete based. So there wasn't a lot of forest, there wasn't really a lot of trees. But what Chicago does have, if you've ever been there, is world-class museums. So I got to go as a 4-year-old to the Field Museum and stare up at these giant elephants. I was there when Sue was unveiled. This is a miraculous place to be a little teeny bopper and just have your nose pressed to the proverbial glass. The Shed Aquarium, if you've ever been, has a whale shark. It's great. I grew up going to the Lincoln Park Zoo. They had this exhibit of chicks that they would hatch from basically eggs all the way to full-fledged chicks, and every time you'd move through a stage of glass, you could basically see this development of chicks, and that was my favorite. I came to find out later that they fed the chicks to the creatures in the zoo, but as a kid, it was really great and really inspiring. Right. So I really liked animals as kind of the take-home. I just was in the wrong place. But thankfully, my dad didn't grow up in a city, so he took us on a lot of backpacking trips. So I did get exposure to trees and the outdoors and camping. And then all of this changed when I moved from this concrete jungle to the foothills of East Tennessee, which if you know anything about the United States, this is like the biodiversity hot. This is like where it's at, especially like salamanders, trillium, darters. So I had a field day. And I didn't really know what I was looking at, but I liked getting dirty. I liked playing in the mud. I liked climbing trees. And so when it came time to make a choice for what I wanted to do for college, I knew that I wanted to study biology. And so I ended up, I told you I've been at Cornell for a minute. So I did my undergrad at Cornell. I knew that I wanted to study biology. But I really thought that if you liked animals and you liked biology, the natural inclination was to be a vet. That was kind of the career path. But there was one little kind of hiccup in that, which is that I really didn't like people. As a vet, you honestly have to deal more with people than you do with animals, and I learned that out. The first time I entered an event, I was like, man, this is a lot of dealing with humans, less than I wanted. But I was really, really fortunate in that my first year at Cornell, I was skimming through the course catalog, and I happened on this class, which was Tropical Field Ecology and Behavior, and I said, this sounds pretty great. So I applied. It was traditionally for kind of upper-level classmen, but I was an over-enthusiastic freshman. So I applied, and I was selected to go. And this completely transformed my life. This was the first time I was able to actually realize that you could study animals in this context and do this for a living. And so it completely kind of shattered my worldview. I will say, I'll admit this, I still wanted to study megafauna. I still was like in on those elephants and really wanted that to be what I focused on. But thankfully somebody else had different ideas. So from that course, I was basically asked to join someone's lab at the Cornell Lab of Ornithology. And again, at the time, I could probably name like 5 birds. And I like, and to the point of like cardinal, not even northern cardinal. But when you're around 300 people pretty much every day who love birds and are really into birds, it's really difficult not to get hype on birds. And so I have fully drank the Kool-Aid, and I am fully on board. As you can see, I'm still doing this 10 years later. So I spent 3 years at the lab, and while I was there, I worked with a lot of different people. I got to see a lot of different science, and this kind of 3-year period was really, really impactful for how I wanted to do my PhD work. So I got to see people who were using citizen science. I got to see people who were putting forth these incredible publications in Living Bird, multimedia. Andy Johnson's actually, that's cute. He's like the first birder I ever met at Cornell. We took a few like sea kayaking together our freshman year. So it's great that we both have kind of stayed with birds. Anyway, so I spent 3 years here. It was really great. It was wonderful. As part of this, I got to study redback fairy wrens in Australia, so I got to do incredible field work in really, really remote parts of the world. My background is in animal behavior, so I started out in kind of reproductive signaling, focusing on that redback. And then, as is the case, field work never goes according to plan, so my study site burned down. So we quickly added fire ecology into that kind of suite of research interests. And yeah, when I wasn't in the field, so I spent 3 years or 3 kind of bouts of field work, I was doing a lot of molecular work, which is how I met Laura, who kindly asked me to speak today. And the important thing is, while I was here, I basically saw this transition in kind of molecular techniques. So we basically shifted from microsats to whole genome data. And this was really, really important for kind of where I saw the frontier of this genomic work going for my dissertation work. I did an honors thesis on sex ratio manipulation in black-throated blue warblers, which is just our females biasing how many sons or daughters they have, and if so, why. Turns out there's like 17 different hypotheses for why, so it was a very, very complicated theoretical project. And I learned a lot from having to wrangle all of these kind of different pieces. And then when I graduated, I knew that I wanted more kind of big data experience, so I went back home and lived with mom and dad. But I did work at the Climate Change Science Institute with Dr. Allison Boyer, and I was basically doing a lot of data crunching to kind of resolve these issues between satellites that revolve around planet Earth and do that and take pictures 2 times a day, but are really, really coarse scales, and then kind of what we need to know on the ground in terms of how these, studying phenology, so kind of like the timing of the rhythm of life. And so lots and lots of data crunching from satellites to basically people measuring trees on the ground. And then I also wanted more nest searching experience, so I picked the easiest nest to find, which is the yellow-billed cuckoo. That's a joke. If you've never had to find them before, they're impossible. So I did a summer of that, and it was pretty brutal. But I learned a lot, and I did get nest searching experience. So when it came time to kind of do the search for where I wanted to begin my dissertation, there were 3 themes that I wanted, which was behavior. So my background was in animal behavior, and I really liked this kind of individual interaction that goes on when we study animal behavior. I also wanted to incorporate genomics because I viewed that as like the frontier where we were going. And then lastly, this element of human change, so how humans are kind of shifting all of this. There are not many people who are doing all of these three things at the time I was looking to go to basically to grad school. But thankfully, I got really lucky. So I think maybe he's spoken at these events before. I heard some people maybe recognize him, but I started working with Dr. Scott Taylor in 2016 at the University of Colorado at Boulder, and I completely shifted focus from behavior to this kind of hybridization speciation framework, which meant I had to learn a lot more TION words, and I had to get a lot better with a pipette and a lot better with computers. So that was a really, really big shift in terms of kind of the theoretical framework for what I was doing. But I really wanted to still kind of keep my roots of animal behavior, and I really, really wanted to kind of bring this human element to science, because I viewed that as really important, that the science that we did wasn't just kept in a filing cabinet or shared at conferences, but instead kind of brought out into the world. So really quickly, I'm going to define hybridization. It's probably the only TION word I'm going to share today, but it's important. So the rough definition I'll give you is the interbreeding of individuals from two populations that are distinguishable by one or more heritable traits. Classic example would be that of a mule, so a horse and a donkey. This is a simplistic barnyard example. And so I think for a long time we thought of hybrids as these kind of fantastical beasts, so this roller bear or this leopard. But hybridization is now pretty well studied, and we know that it's really common in natural wild systems. This is hybridizing canids. Anyone fishes salmonids, especially out west, they hybridize like mad. And then of course birds are really, really, really well known for hybridizing readily. So Scott had just come off of studying these black-capped and Carolina chickadees that hybridize out east. And so when he came to Colorado, that was kind of the framework that he wanted to keep expanding. So in the framework of basically Colorado, he said, okay, let's study hybridization, but this time we're going to do it with black-capped and mountain chickadees. So the first thing I did was scan the literature, because I was a good student. Sorry. I want to briefly pause and say, sorry, there's a quick aside. So I was Scott's first student, which was great in many ways, but it also meant I was the first pancake, which if you know anything about making pancakes, it's that sometimes they can be a little deady. I think I did okay. But this was a really beautiful opportunity because I had an entirely blank slate. There was nothing. We had no permits, we had no lab, we had no pipette. We had a dream of studying chickadees and studying hybridization, but you can do that a lot of different ways. So again, he gave me this system, black-capped and mountain chickadees, and kind of told me to run with it. And I had a great time doing that. Okay, so I scanned the literature because I was a good student, found this, basically there are three things that pop up, one of which was from Colorado, which was really exciting because again, we're in Colorado, that documented this actual social pair between a female mountain chickadee and a male black-capped chickadee. And they had these really, really nice film photos from, again, this is 1996. And so you can see it looks pretty black-cappy, but maybe has like a really faint white supercilium. So this was encouraging that maybe we were onto something. These birds don't readily hybridize, but maybe they hybridize sometimes. So the next thing I did was take all the reports from eBird of where people had seen hybrids and plot them on a map. And I'll mention briefly, black-caps are going to be shown in pink through the rest of the talk. Mountains are going to be shown in blue for the rest of the talk, and then anywhere they hybridize will be shown basically in purple, or in this case where their ranges overlap. So if you want a nice, that's a side-by-side pic. We caught them at the same place in Colorado, so they do overlap. Key differences in what they look like is, again, that white eyebrow of the mountain chickadee, and then the white wing bars coming down the wing of that black-capped chickadee. I think I have a laser on this. Oh yeah, okay. Eyebrow, wing bars. Cool. Okay, so I went onto eBird and found pictures of birds that look like this. So you can see both of those characters. There's that white supercilium, that white eyebrow, and then also those white wing bars. So this is something that looks half like a mountain, half like a black-cap. And then I plotted a map of where I saw all those. And if you squint, which is exactly what I did, which is the scientific way of evaluating anything, you can pull out a couple of major urban centers on this map, right? You can see Albuquerque, New Mexico. There's the Front Range of Colorado, Cheyenne, Wyoming, Salt Lake City. So it seemed like maybe there was something going on with cities that was potentially having something to do with where these birds hybridize. And when I contrasted it again, so we know that blackcaps and Carolina chickadees readily hybridize. And the key thing here is that they hybridize everywhere they come into contact, right? Everywhere along the zone from Kansas all the way to New Jersey. Anytime blackcaps and Carolinas overlap, they hybridize. That's not what we think at the time was happening here. So it seemed like there was something context dependent about the hybridization with blackcaps and mountain chickadees. And it seemed like the context was people. So that led me to a lot of digging. But again, humans are pretty dominant forces on Earth. We really transform landscapes. We completely transform ecosystems. And so I did a lot of digging on this. Could there be something that humans are doing to the habitat that's promoting hybridization? Turns out someone else had this idea back in 1948, so it wasn't just me. But they were studying spiderworts in Missouri Botanical Gardens, and they found basically one species on sunny slopes, one species on kind of like these wetter, shadier slopes, and anywhere where people had modified the habitat, this guy, Anderson, found hybrid spiderwort colonies. And so he postulated that humans could do something to the habitat such that hybridization was more likely to occur, or hybrids were more likely to survive. So again, this isn't a novel concept, but what is novel is our ability to detect it with genomic data, right? So now we no longer have to rely on how things look, if they look like a hybrid, to be able to identify hybrids. Okay, so I'm going to call this, sorry I told you it wouldn't be too jargony, but disturbance-mediated hybridization. I'll illustrate it schematically. We start with two species that exist in the same place, here bluebirds and yellowbirds. A disturbance occurs, in this cute schematic, a city is built, and now, or we didn't prior, we detect hybrids. So that's basically the concept of what we think is going on. And so I went back to the literature, did lots of reading, and found a whole bunch of examples where this was happening in a lot of different taxa. And then put forth this review, basically documenting this pattern widespread geographically, and also in a lot of different species of organisms, including birds. But this left one really key question, which is how are humans doing this? What about these disturbances are actually causing this hybridization? And that is what I spent the bulk of my dissertation work exploring. So this is, don't read this text, this is a terrible figure. This is like the first figure I ever made. And the key point is that there's too many words. But the big one is up here at the top. If you wanted to actually study this, you would first observe hybridization. Then you would figure out how long hybridization has been going on. So does it track with when people have been modifying the habitat? And then, because you're a good scientist, you would conduct experiments. So you would figure out what mechanisms. I'm going to spoil my talk and say I really only got to the first 1.5 because this takes a really long time and science is really hard. Okay, so we're going to focus on the first one, which is observe hybridization. So again, we have this report of where we think birds are. So these are birds that just look like hybrids based on phenotype. But I was curious if these birds were actually hybridizing in places more disturbed by people. So the first step, really simple, is just to say, do we see hybridization relating to these habitat disturbances? This is really quickly one to acknowledge. I didn't collect any of these samples. These are chickadees that were caught by Dr. Teresa Berg at the University of Lethbridge and Dr. Ken Otter at UNBC. So just want to acknowledge, I did all of the lab work and other things, but none of this would have been possible without them driving across the country to collect birds. Am I done? Am I out of time? No. Okay, so step one is measure disturbance. Thankfully, because I worked at the Climate Change Science Institute, I knew a lot of people who had access to a lot of really great data sets on land cover. So this is a really fancy kind of composite of a whole bunch of different measures of how people have changed the environment that scales from 0 to 64. Then we plot all of the birds. I plot all the birds that basically have been sampled there and then extract that value for how disturbed that sampling location is. And then the next thing is measure hybridization. So this is done using basically pretty low coverage sequencing. The key point here is that this is cheap, so it allows us to sample hundreds and hundreds of chickadees. And then this is what it looks like when you take 500 individuals and shove them into one tube, basically. There's a lot of clear liquid being passed between vials. It can be a little stressful, but ultimately this is what gets sent to the sequencing facility after months and months and months and months of work to deal with hundreds and hundreds of samples. So that's what we're doing, is just comparing disturbance to hybridization. And I'm going to briefly talk about a metric that I'm going to use to measure hybridization because I'm going to talk about it later in the talk. And that's called a hybrid index. So it's just an index of how hybridy you are. It's going to scale from 0 to 1. So again, our bluebirds to our yellowbirds. hypothetically, if these bluebirds and yellowbirds had offspring, we would call that an F1 or filial, that's what the F comes from. That's going to have a hybrid index of 0.5, so right in the middle. And then if this F1 hypothetically breeds back in the direction of the yellowbirds, we would call that backcrossing. And then that would produce a backcross that has this hybrid index, again, on average of 0.75, and if it goes in the other direction, 0.25. So just a really simple metric of how hybridy or how mixed up the genomes of these birds are. Okay, what I found really excitingly is that we do see a correlation of disturbance and hybrid index. So again, this is just how disturbed that habitat is on the X, and then that hybrid index that I just showed you is on the Y. This one is only going from 0 to 0.5 because I scaled it to make it much easier to plot. So this is literally just for the ease of looking at it. But we see a really nice positive significant correlation, which is great. Maybe not if you're a chickadee, but anyway. Okay, then where are these birds hybridizing? So plotting basically these birds on the landscape, we can see that they're hybridizing in a lot of places, so it's pretty widespread. Okay, so did I observe hybridization? Yes, cool. Remember I told you we're only getting like 1.5, so sorry. Next is to quantify hybridization. To do that, we're again really interested in how these disturbances are promoting hybridization, and that requires two things, in my view. First is we need to understand what these birds are doing, right? We need detailed field studies to understand on the ground how these birds are living their life, what decisions they're making, and then lastly, quantify hybridization, which requires genomic information. Okay, so I took this map that I plotted with all these birds from all across North America, and then I said, now what happens if we take these birds from eBird that I showed you earlier? Do we see any overlap? And lucky for me, remember I'm at the University of Colorado Boulder while I'm doing all this, but the Front Range of Colorado pops up as being pretty much a hotspot. So great, now we're going to figure out if chickadees are hybridizing in Boulder, and if so, why. And if you've never been to Boulder, Boulder is a pretty unique place. You basically have the grasslands of kind of the middle of the country tram up right against the start of the Rockies. And this is, I told you I was going to show some heavily edited photos. This is not actually what this looks like. But the key point basically is you go from grasslands to really, really big mountains really fast. It also is unique in that it is a city, like a pretty solid city, that's surrounded almost entirely by protected green space. You have this really beautiful gradient of urban and rural that's legislated and protected, which in theory would set up a really nice opportunity to test some of these questions of, are cities doing something to how chickadees are interacting? Okay, so now I took my question and made it much smaller, which is, do chickadees hybridize in Boulder? And if so, why? With the aim of monitoring wild chickadees and then measuring hybridization. So pretty simple. This seems so small. This is all you have to do to get a PhD. So it's great. Okay. So first, measuring hybridization in chickadees. To do that, you need access to the actual birds. We collect basically blood from the brachial vein, which requires catching these birds. We also want blood from their chicks. If you've ever tried to reach your hand into a chickadee cavity hole, which I have, let me tell you, it doesn't get very far, and you can't actually access the chicks. But, like for us, you can build these really nice artificial cavities that they will readily nest in. So this was the plan. Basically, this was the plan for in year 2 of my PhD, was I'm going to build a bunch of boxes, and we're going to get wild chickadees to nest in them, and then we're going to measure hybridization. So I did that, which involved driving up to this really cute little wood shop, because nobody on campus would let me use a saw, even though I told them I knew what I was doing. Then I spent hours, so many weekends, cutting wood, loading up my car, not my truck, I wish I had a truck, loading up my CRV, driving them back down to the lab and building hundreds and hundreds of boxes. The first box I built took me probably about 5 hours. By the time I was done, I could do it in 10 minutes. So I got pretty good. You build the boxes, but of course there's lots of things we want to consider. So they get this nice little tooth bender on the front, which I really like this photo because it shows that they work. So this is to prevent anything from expanding that hole. That hole is capped at about 2 inches, like 1 3/4 inches. So it's meant just for chickadees. We do get other things in there, but they keeps out squirrels, which is nice. The other great thing that I did was decided that if we were going to make this project legitimate, we needed a logo. So that was really step 1. And this is, I think, the beauty of combining art with science. So I did not make this, but I did have some intellectual input. But I had the great idea when I was building all these boxes is, man, I always see nest boxes up around, and I want to know what they're trying to attract. What if we put a logo on the side of the box? And then people could figure out what we're trying to attract. And they serve as mini billboards, because the plan was to put this all over the county. So thankfully, when I was in 6th grade, I took a screen printing class. So I knew that this is not that hard. Anybody could do this. You can go to like Michael's and get stuff. You just need a really bright light in a dark room, which thankfully in a cell lab, basically we did have dark rooms. So they hold up pretty well. They turned out pretty okay. Okay. So this is a really terrible map. I didn't make it, but what I want you to pay attention to is that this is the city of Boulder, and then all that green stuff is protected. It's what's called open space, but it's protected parks that are regulated by the city of Boulder. And the idea originally when I built all these boxes was we were going to put some in the city and some outside of the city to compare what's going on in the city and what's going on outside of the city. To do that, we had to go to the city of Boulder and then also open space and mountain parks and ask them for permits. I did this for 2 years. I had so many meetings, I can't even count them, and at the end of 2 years, they told us no. Right. Which was pretty devastating because this is a really beautiful experimental setup of rural versus urban that is really, really unique to Boulder. But it was a really great lesson in bureaucracy and also changing gears. So I decided, okay, fine, let's just put these in people's backyards. And at the time, that sounded pretty easy, but it was not. So I had to figure out how many people, how was I going to get people? So the first thing I did, thankfully I had this nice logo. Again, this is again a key, I think the reason this project is successful is because I had a logo. Honestly. So I put out this call on the Colorado Birds in the winter of 2019 asking people to join this study, to host boxes, to help monitor these boxes, so that we could learn more about what's happening with basically chickadees in Colorado. And then I started emailing everybody that I knew that had a house, which in Boulder is a smaller number than you would hope because having a house in Boulder is really difficult because it's expensive. But I thankfully knew people who had been in Boulder for a really long time, and they knew people who knew people who knew people who knew people. And that is really how I got this project off the ground. The other thing I want to point out is that carrying these boxes around, basically mountainous terrain is really, like, a person can really only hold 2 at a time. And I put out just shy of 400. So carrying these things over mountains is an incredible logistical feat that I was only able to accomplish because I had a lot of people who are willing to help. So we put up these boxes, we keep track of what number goes where, we take lots of measurements, how high they are, what direction they take. This is what I do when I give the real science talk, but the actual reality is, again, it's really difficult to move all of these boxes on this little sled. That's pretty much all we had. I ended up buying like a child sled for like kids, but this one was better. The other thing is this is like death by paper cuts. What tree do you pick? How high do you put it? Which way do you face it? And I had no idea what I was doing. I read, what literature there is, but a lot of this was just kind of me guessing like, that looks good. If I was a bird, I'd go there. We really didn't know this was going to work. This is a huge gamble. I've spent over a year building these boxes, and then I spent a year putting them out on people's properties all throughout Colorado, and we didn't know if it was going to work. This is my birthday. This is a great email to get on my birthday. Not all of this was happy. So some people were really upset about the boxes. This was I thought a long time about this email, and I put it in here to say that working with people is a joy, but it also sometimes makes you really evaluate why you're doing things. This one particularly stuck with me, that I was luring these birds to their death. And I did for a really long time think about my ethical responsibility as a researcher. Is that what I was doing? I can now tell you no, we really don't have much mortality, and I ended up not working with this woman because they had a lot of opinions. But this is what happened after 2 years. So this is 408 boxes. I showed this picture like so quickly in talks, and it's really nice to be able to talk about the amount of effort it took to make this map. This is years of labor to build these boxes and put them up and pick where to put them. But this is what it looks like from a bird's eye view. To do this, again, I had to work with a ton of people. So I worked with 4 departments, 3 institutes, 3 campus units. I had at the time, smart idea, I guess, of putting boxes on campus. But it's not really, there's no one to ask if you can do that. What are you going to do? Go to the president and ask them if you can put boxes on a university campus? You have to talk to one person who sends you to another person, who sends you to another person, who sends you to another person. So I did it. I ended up, the person who actually matters is the head arborist, if you ever need to know how you need to get boxes on campus. I ended up recruiting 75 people. So each one of those people I basically cold called either on the phone or through email, or basically meeting them in person and sat down with them. I had tea and coffee and explained what I wanted to do. So that's 75 plus hours just getting people to agree to this, not including people that I didn't end up working with. And then I also worked with middle schoolers, high school students, community college students, and undergraduates to build these boxes, put them up, and then ultimately monitor the birds that lived inside of them. So this is an incredible labor of love, and it's been really beautiful to work with so many people. I think this is what comes back to me growing up in a city, is that this in some way was like only a city girl kind of dealt with this many people, even though I didn't want to do that. So I just want to acknowledge lots and lots and lots of people who helped me put up these boxes. These are all the people that let us come onto their property for basically 2 months out of the year and keep track of all their chickadees. And then this is just lots of pictures of me working with many, many people. The kids like to sign the bottom of their boxes, which I always think is great. Yeah, that was always really fun. Turns out I take like 10 minutes to build a box with a drill. They take like 2 hours, but it's good. It's good for them. Okay, so back to a little bit more science. This is a picture of basically all the boxes that we have in Boulder County. So they start down in Boulder. It's about 5,000 feet. Again, these are just boxes in people's backyards. Pretty standard. Also boxes on campus, as I mentioned, so that's that giant cluster. And the goal here was to incorporate this into classes and also to help students who didn't have cars, who wouldn't be able to access this really montane environment. So that was that. Next is this mid-elevation site, so about 6,000 to 8,000 feet. This is Sugarloaf is the name of that property because that's the road. This kind of gets more like open ponderosa, some aspen groves. This is what you drive up a dirt track that can be dangerous in the freeze-thaw cycle, but you get really great views of the mountains. There's lots and lots of mining in this part of the country, so I learned pretty quickly when I was going onto people's property to ask where the mine shafts were, so I didn't fall in them. This is an old tungsten mine. Yeah. So that's the mid-elevation, and then all the way up at our upper elevation site, these boxes go up to tree line. So this is tree line, these little stubby trees, and we get mountain chickadees that breed all the way up here. So really beautiful views, and again, mostly spruce and fir that are up here at this elevation. So completely different habitats. The key point here is that they span kind of this low elevation, oh that's like purple, that's supposed to be pink, but this low elevation habitat of blackcap and then this high elevation habitat of mountains. So we're spanning the elevational distribution of both of these birds, ideally figuring out where they interact and overlap and if they hybridize. So around the time that the first snow begins to fall, which is like the middle of April, we start monitoring boxes. Sometimes it's easier said than done. Every site has its challenges. So on campus, sometimes people like to make it a little difficult for us to get our boxes. This was a nice little fresh cougar kill that I found at that mid-elevation site. And then up at high elevation, there's lots and lots of snow. So sometimes getting down to the boxes in these snow wells can be challenging. We also don't always get chickadees, so we get unexpected visitors like this cute little snake, we get hornets, we get all sorts of different types of birds. But when it does go right, and we do get chickadees, they start excavating the box. We fill them with shavings so they feel like they're doing something. They're primary excavators, so they have to kind of pull out those shavings to use the boxes. They start by, the other point I'll really quickly point out is by working with citizen scientists, they take really great photos for me and then allow me to use them. So none of these are mine, but the vast majority of them were taken by landowners who hosted these boxes. So this is a blackcap nest, starts pretty mossy. I like this little action shot. They're very, 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 very industrious. They can basically excavate and build in about a week, which is pretty crazy. And then they start laying eggs, which are sometimes hard to find. So there's one in there. They'll cover them until they start incubating, and once the female incubates, she'll remove kind of that fur cap. Here's a nice side-by-side comparison of blackcap versus mountain. So mountains tend to be just overall fluffier. They have more fur. They also lay bigger clutches, and their eggs are pure white as opposed to having that kind of speckling. We call this the gummy bear stage. This is freshly hatched. Yep, it's pretty. They're like day 1, day 0, day 1. And then we monitor them until they're day 12, so it gets to be pretty crowded, but that couple will expand with them, which is really beautiful. So we check these boxes. When they're day 12, we pull out the chicks. We do all sorts of pretty standard science stuff, which involves measuring them, weighing them. The chicks get a little metal band so we can keep track of who's who. Again, lots of measurements. Sometimes they poop. We collect their poop to figure out what they're eating. And I used to catch birds with mist nets, but it turns out chickadees are not very producible to that. So instead, you just wait for the parents to come back, and then you plug the hole and then reach in the top. So it's a little bit of a different way of trapping birds, but slightly more effective. For the adults, we give them color bands, so this kind of unique plastic colored bracelets so we can keep track of who's who. And then we do the exact same thing we do for the chicks. So we collect blood, we take measurements, we weigh them, take photos, and then we also conduct provisioning surveys so we can figure out who's bringing food to the nest, how often, etc. And then again, these boxes are all used on campus. They're predominantly monitored by undergraduates or landowners, so I really only monitor a very small fraction of them. This is just a little snapshot of folks that I've worked with, just to highlight all their efforts. And so, in general, what we collect is the day that egg is first laid, how big the clutch is, when those eggs hatch, how many chicks do they have. We, of course, collect genomic information from parents and adults, and then we also look at feeding rate. There's a bunch of other stuff we collect, but this is kind of the highlight of what matters. And again, I just want to acknowledge that I would not be able to present this data without the work of a lot of people who've helped collect this data in the field for the past, at this point, 6 years. Okay, so I said 373, it's actually 408, and then monitored 127 nests. I'm going to show you only data from 3 years, because that basically is what I had for my dissertation. Banded 203 adults, and then there are 274 associated nestlings. And so again, ultimately what I'm interested in asking or answering is, are chickadees hybridizing? And then what about their ecology either lends them to hybridizing, and then what are the consequences of that hybridization? So really surprisingly, what I found is that these birds overlap a ton. So when I first started this project, I would have told you that blackcaps were down in the city and mountain chickadees were up in the mountains, and that's not actually the case. So this is now, instead of boxes, this is now nests. So places where we actually monitored nests. Pink, again, is blackcaps, blue is mountains. And we have mountains that breed down in the city of Boulder, right next to neighboring blackcaps. And we have blackcaps that breed all the way up, pretty much almost to our highest boxes. So way higher than I would have anticipated from the literature, from any sort of anecdotal experience. Shown in other ways, this is that same kind of like elevational pie graph. I'm just going to basically stack nests on top. And so you again can see that we have mountains breeding down in the city of Boulder, and then blackcaps that breed all the way up, almost to our highest elevation site. So they overlap a ton in where they breed. They also overlap in when they breed. So on the x-axis here is this date, and then on the y is going to be the number of nests that had their first egg laid on that date. For blackcaps, that's about the end to first week of May, and then for mountains, that shifts to about the middle of May. But again, a lot of overlap in when these birds are breeding. So they're breeding in the same place at the same time, which means there's the opportunity for hybridization. But do they hybridize? And similarly, this holds if you basically take that same graph and break it out into 3 years, because people care about that. Still holds, and it also holds if you look at when those eggs hatch instead of when that first egg is laid. So this pattern seems to be good. Okay, do they hybridize? This is the drum roll. To do this requires a lot of lab work. Sometimes it looks not very glamorous, but it looks like this. Schematically, what's happening is we're taking blood from birds. Birds have nucleated red blood cells. We do not, but birds do, so they have DNA in their blood. We then basically add in a whole bunch of salt and alcohol and do a lot of really amazing chemistry that other people figured out in the 90s to extract DNA. Then we do a bunch more fancy chemistry. This part involves lasers. So then basically this is sent to a sequencing machine. They shine some lasers on the DNA, and you get this crazy grid, and then ultimately that gets translated by a computer to ultimately what we get, which is a bunch of A's, T's, C's, and G's. And then we take the A's, T's, C's, and G's and put it through a whole bunch more computational work and a much larger computational pipeline to then actually get those metrics, one of which is that hybrid index that I showed you earlier. Some days in the lab are good. Some days are not. This is okay, they were numbered. So they got to go back, but I just took a photo instead of crying. But it's just a reminder that some days are good and some days are not good. So again, these are genomic samples that are coming from, all of these are nests that I'm showing you. So each one of those nests is going to have 2 adults and then about 6 to 8 chicks. And so again, all the genomic information from those individuals. And what we find is that they do hybridize. It's pretty rare, and then we do see some trends to the hybridization going back in the direction of blackcaps. What this means here is there's 500 individuals, and this is 143 places in the genome that this information is coming from. What I'm going to show you is what's called a triangle plot, because it's shaped like a triangle. Hybrid index is going to be on the X, which is that same metric that I showed you, which is how hybridy you are. And then heterozygosity, which is, I know, a big, like, kind of crazy, but it just means how mismatched your alleles are across your genome. So you get one copy from mom, one copy from dad. You want to know how mismatched those copies are, and that's what that metric is. The big thing is that blackcaps are going to be down in this corner, mountains are going to be down in this corner, and those F1s, that first time that those blackcaps and mountains cross, will be up at the top of the triangle. So this is the schematic. What I actually found is this. So we found one F1. This is a female that looks like this. You can see she has this faint supercilium. And other really weird things involve constructing a nest that lacks a fur cup. So there should be that kind of bundle of fur that I showed you. There's no bundle of fur, so her nest is not done. She then didn't lay any eggs and then incubated the bottom of this box for 2 weeks before abandoning. So pretty aberrant, maybe not necessarily the most we would say reproductively successful, but she did pair with a male and she did construct a partial nest. So they do hybridize. We found one F1. Granted, not a lot, but one. So yeah, that's supercilium, emptiness, etc. These individuals are also really exciting. So they fall in kind of that trend of potentially being something that's not a pure parental, but not a hybrid or an F1, but something in between. We call these backcrosses. So I ran those individuals through a bunch of simulations. I'm going to show a really quick schematic. What's going on here is these vertical bars are individuals, and then the proportion of the bar that's colored is going to correspond to which genotypic class they assign to. So this individual would assign predominantly to blackcap with a little bit of this kind of backcross class, and then this individual would assign perfectly to being a mountain. So this is just using a bunch of computers to validate what we think is happening with this kind of simple metric. So that's the sample schematic. This is the actual data, which I know looks like a rug, but that just means our simulations are really good and that everything perfectly assigns. Key thing here is that we see those hybrid individuals perfectly assigned to that backcross category. So these are second generation hybrids. So they're F1s that have basically bred back with those pure parentals and then produced these backcross individuals. And because we have boxes, we know, this is the cool thing, we know who all these individuals are. We know where they bred, we know who they paired with, we know how many chicks they had. This is the benefit of basically building all those boxes. We haven't gone out to sample chickadees and then gone back to the lab to run genomics. Instead, we know how these birds have lived their lives, which is really powerful. Okay, the other really cool point that I want to lastly point out is that these are the rest of all the birds that we sampled. And all of these birds are elevated away from zero. So they should be down here where basically these solid dots are, but they're not. They're shifted away from zero. And what this suggests is that every single chickadee in Colorado, at least in Boulder County, contains some piece of the other species genome, which means that either hybridization has been going on for a really long time, or lots of hybridization has been ongoing recently. Either which is pretty exciting because we didn't know that these birds hybridized when I first started my PhD. Remember, there were 3 papers that said that they hybridized. Okay, so another way of showing this is that exact same thing. You can see that these clusters are elevated away from these ones, so they don't overlap, suggesting again that they have a different amount of ancestry from the other species. Okay, where did we find hybrids? So remember I showed you that we sampled both species all across the elevational distribution. So mountains down in the city, blackcaps all the way down low, all the way up high. Where are we seeing hybrids? And we see them sampled at low elevations, predominantly in the city of Boulder. This is like a one-off. This is a female that we just happened to catch while basically setting up a net. We didn't have information for her nest or who she was paired with, so which is interesting. But everybody else was basically sampled down in the city of Boulder, which suggests that potentially that's where hybridization is happening in the city as opposed to in the mountains. Okay, and the other thing is most of these birds that are backcrosses are males, which is also important. Remember I told you that other bird that was an F1 was a female. And so this is support for Haldane's rule. So Haldane's rule is one of the few rules in biology that's been upheld. Most of the rules in biology tend to be broken with exceptions, but this one seems to be pretty good, even though it was put out 100 years ago. But it says that in hybrids, if one sex is absent, rare, or sterile, it's the heterogametic sex. And in birds, that's females. So this female, again, would be the heterogametic sex. And so she did not lay eggs. She built an incomplete nest, so pretty sterile, if you would. As opposed to those other individuals that I showed you that were all males. So, briefly, chickadees overlap in when and where they breed. Even though they overlap a lot, hybridization is still really rare, but we see those hybrids being produced in the directionality of being basically produced back towards blackcaps. and they're produced at these low elevation habitats, i.e. in the city. So I told you I was going to maybe ask how, but again, science takes a long time, so I haven't gotten there. I mostly have just shown that they do hybridize, which is great, and the next step is figuring out why. Remember I told you I'd get to like 1.5. So this is what I'm working on now for my postdoc, which is thinking about why these birds are hybridizing. We know that mountain chickadees undergo these kind of sporadic elevational movements, which has been documented. We don't really understand why. And the other thing is this is what Boulder used to look like in the late 1800s. It was a pretty quaint mining town, and the big key takeaway is that there are no trees. Now this is what Boulder looks like. It's incredibly deciduous and tree dominated, and who really likes trees, deciduous trees? Black-capped chickadees. Like birds, yes, but black-capped chickadees. So we think potentially what's going on is that basically Boulder has become this kind of bastion of blackcaps that have been artificially increased by people planting these really nice ornamental deciduous trees that shouldn't be there. And then these mountain chickadees come down from these high elevation habitats and then interact with blackcaps down low, and then that makes something happen that causes them to hybridize. But the key thing is that it's kind of extrapolating these impacts at the city elevation kind of ecosystem level to consequences at the genetic level. And with that, that's the end. Lots of people to thank. Of course, collaborators. Couldn't have done this without a lot of people. It takes a village. And people who gave me money, who saw value in the work that I was doing. And with that, I'll leave that up and I'll take questions.
So you were talking about the hybridization of the birds on the west coast, or Colorado, and but you said also it was like in the Blue Ridge Mountains, you had that line where, and that's not city at all.
Yeah. So she was asking about, you're asking about this one?
No, yeah.
Yeah. So this is blackcaps, which is interesting. So one of the players in these scenarios is the same. So this is blackcaps. These are Carolina chickadees. So it's a different species, and the key thing is that everywhere that those ranges come into contact, those birds hybridize. That hybrid zone is really well studied, super famous. Lots of people have done a lot of work on it. This is blackcaps, which is the same, so that same kind of northern player, but now we're looking at mountains, which are these kind of high elevation coniferous
So that has
specialists.
nothing to do with city over there.
To me, the key thing is the pattern is different, right? The key thing there is the pattern is basically everywhere those birds come into contact, they hybridize, which suggests that it's something about how they interact together that's causing them to hybridize. Whereas these birds overlap a ton, right? Like there's tons and tons of purple, but we only see, granted, like take this with a grain of salt, observer bias, etc., but we only see hybrids being reported in some places. So they hybridize sometimes, but not all the times. Whereas these birds seem to hybridize all the time. And that's kind of the original observation that was like, is this people? We still don't really know, but it looks like.
And one of the differences is the species.
Yeah, exactly, yes. The blackcap is the same. So both of these involve, this is blackcap, and then this is blackcap, this pink. But this one is Carolina, and this one is mountain. And Carolina and blackcap are sister taxa, so they're more evolutionarily related. So it would be expected that they would hybridize more readily than blackcap and mountain. Yep.
So you expect any changes in vocalizations among the various elements?
Yes. Okay. Great question. The answer is yes. Anecdotally, we have recordings of birds that look like one and sing like the other. In those cases, we haven't. They're not necessarily the birds that are using our boxes, so we don't have genomic information from them, so we can't compare vocalizations to genotype, which would be great. But I just basically reviewed a paper that I've been working on with one of the undergrads that I showed up on that slide, and she was looking at song. And what we do see that's really, really interesting is that mountain chickadees in Colorado sing very, very differently than mountain chickadees elsewhere. Such that when I first was doing this fieldwork, I was listening for that drop in pitch, and they don't drop. They just go dee, like dee, dee, dee. There's no pitch drop. They sing completely differently.
So that's going to ruin Merlin.
It just demonstrates how difficult the Merlin problem is, that you basically have to account for a lot of variation across a really, really, really broad spatial scale. But yeah, so we do have evidence that they sing differently when both of them overlap, presumably so that they don't hybridize, right? Song is supposed to be a mechanism that prevents you from breeding with the wrong species. But yeah, you had one, yes.
Do all the female hybridized birds, are they all sterile or?
So we only have the one, and I know it's just the one, I know. So I just wrote up this paper, and it's like, albeit small sample sizes, IN of 1. So yeah, just that one, but that one is pretty sterile. I mean, like, she had a brood patch, but she didn't lay any eggs. And the other thing, the crazy thing, is that incomplete nest. That to me is really striking, that nest is half done. It's lacking, and it's half done in a way that's like very easy to compartmentalize. Like, you build the moss layer, and then you build the fur cup, and the fur cup is just gone. Yeah.
Wouldn't that be like a first year mama? And
Well, we know that she's a hybrid because we genotyped her. So she could be age dependent, but we would like to presume that it's because she's a hybrid. And we know that she's an F1. Yeah. There was some other, we're just gonna,
Can you repeat the questions?
Oh, yes, totally. Sorry. Do you want me to go all the way? The first one. How good is my memory? Not as good as the chickadees. Yeah.
Until a few years ago, I grew up in Berks County, Pennsylvania, which is right over that white line, or the yellow line. And over the years, we saw the Carolina come up and hybridize with the black caps. That line of hybridization keeps moving north. And we know that the Carolinas learned the song of the blackcaps to lure them in. And the Carolinas are a more aggressive bird. Did you notice a difference in aggression between mountains and blackcaps?
Oh my gosh. I'm like, lucky for you, I learned from someone who's really good at giving talks. So I have slides prepared. We have not done this work in Colorado, so this is one of the things. Chickadees are really well studied, which is great because it means I can answer a lot of these questions by going to the literature. But people do know dominance matters for reproductive decisions, so females typically pair with more dominant males. In blackcaps, the more dominant birds tend to be older males. And when you lump blackcaps with mountains, blackcaps are almost always more dominant over mountains. And this is really nice because it validates the directionality of this hybridization, that the hybrids are breeding back with blackcaps. And also, if you remember that original thing that I threw up, I was like, I should have these all together, but that original publication from Colorado in 1996, that also is basically paired, the pairing is the same. So it's a female mountain pairing with a male blackcap, which is what you would expect given those interactions. So yes, there are dominance differences. We assume that they matter, but again, I built all those, but I was out. Someone else is doing that now, but that's where people are looking at now, is kind of validating, do those dominance interactions stand and do they matter for reproductive decisions? Yeah.
Could the urban environment possibly lead to the creation of a new species? Because even if there is going back, you're different from the original chickadee. And the environment is another factor which provides some driving force for development, so they're not all sterile. So usually we think of urban environments hindering speciation by eradicating species. Could it go the other way?
Yes, I first am going to touch on your point as urban environments hindering speciation, because I think that's, if I had to encapsulate all of my work, I would say we think about when people build cities, they cut down trees and birds can't live there, but we don't think about you've planted trees and now you've changed how things in those environments interact. So this is a very different way of thinking about human change, and that we're not changing where things are or how much things are there, but kind of the status quo or the social norms of how things are existing or navigating these urban environments, one of which of those being hybridization. Your original question of whether or not these cities are going to cause these birds to form another species, probably not. We typically only really see kind of hybrids becoming, so in order for that to happen, they'd have to form hybrids, and the hybrids would have to only mate with other hybrids and not the other species, or if they do breed with the other species, it would have to be a really, really high, basically they would have to not successfully reproduce. And that doesn't really seem to be the case in most birds. It's common in plants. Plants are a pretty good example of that happening. But it's probably not going to happen because I just, that F1, right? Like, she's not reproducing. So already that F1 can't reproduce, so it's probably not going to form another species. What is possible is potentially kind of shifting, this is like a very nebulous term, but shifting the like adaptive potential or evolutionary trajectories of these birds. So there's something about this influx of mountain DNA into blackcaps and this influx of blackcap DNA into mountains that maybe would make them better at navigating warmer environments, make them more dominant, et cetera, et cetera. So it might change their ability to respond to change, but it's probably not going to make them a separate species.
In your triangle graph, about how many were those pinks versus the reds?
Oh, it's like I could just pull them.
What's the question?
Oh, on my triangle graph, how many of the pink squares were pink squares versus red? That, yeah, okay. So basically this versus this and this versus this. Yeah, so these dark circles, those are birds from places where they're called allopatric populations, so places where we only have either blackcaps or only have either mountains, the idea being that that's a good representation of what's pure or not going to be influenced by hybridization. These ones come from California, and then these ones come from Kansas, so basically pretty close to where we're sampling. And there's about 15 in either of those clumps. They're just stacked right on top of each other, so you can't see them. They're it's like they're transparent in the way that like that is transparent, but because there's so many and they're all stacked, they look like one point. But there's 15.
So there's about like 3 pinks for the 15 red, approximately.
Sure. Yeah. Mm-hmm. Yeah. Yeah. And there is and the other thing that's interesting that I don't really talk about this, but the variation is greater in blackcaps than it is in mountains. These mountains are really, really tight around each other, and there's way more variation. And when we make this same plot with that original, remember I showed you that original graph of comparing disturbance to hybridization, those are completely different birds. They're just randomly sampled across North America. And this was like a really very consoling to me. The graphs look pretty similar. I was like, okay, so we have 2 different data sets, we have 2 different sequencing technologies, and they're still showing the same patterns, which is good. That's a good validation. Because I always worry I've done it wrong.
Which is your favorite?
Oh, none of them. All of them. I think the other thing I really love about chickadees is everybody knows them pretty much. Even if you don't know birds, you're like, it's the Christmas card bird, so it makes it really easy to relate to people. It's a common tattoo bird also. And I think it's really beautiful that these are really, really common, widespread, well-studied, and we're still learning stuff about them. And people ask me questions all the time, like, how do you figure out where to put a box? Like, you'd think that's a simple question, like, where do you put a box to attract a chickadee? It turns out it probably doesn't really matter because they use the boxes pretty well. But I think it's beautiful that we have a lot left to learn from backyard birds that we kind of one off and considered common. So they're one of them, yeah.
So what was the utilization of the boxes?
Oh man. So it's biased because I basically was like, I don't know if this is going to work. You have to remember, I'm putting all these boxes up in 1 year, and I'm like throwing all my proverbial eggs in one basket, hoping that this project pans out so I can graduate and get a PhD. That's the plan. So when I would go to a property, I would put up multiple boxes in a yard, being like, okay, this is 1 yard. I'm going to come here, and I want to basically maximize my ability to get birds to nest in these. So in any given yard in the City of Boulder, most yards had like 2 to 4, depending on the yard size, but only one of those boxes would be utilized in a given year. On average, we had about 30 to 40 successful nests every year. So that means that they laid eggs and we were able to measure them at day 12. Failure rate, they do fail. So some years they fail either because there's snow or because house wrens come in and outcompete. We now have a project on house wrens because they like our boxes so much. So we just have co-opted them to study something else. But occupancy is pretty high, like 30% if I had to say. I mean, I'm like spitballing, but pretty good. Yeah. So if you want to know how to attract a chickadee, I got building plans. I can tell you how to do it. I'm not doing it. I'm done.
Well, thank you very much.